It's my pleasure to introduce Professor Muzlifah Haniffa for the second keynote of the day. She is a clinician scientist, a professor of dermatology and immunology at Newcastle University, and currently the interim head of Wellcome Sanger Institute. As a champion of international scientific collaboration, Muz is one of the pioneers behind the Human Cell Atlas, which is a global initiative to chart all the cells in the human body. Her research has transformed our understanding of how the human immune system develops and how cellular dynamics change in skin diseases and leukemias. Her work has been awarded the Academy of Medical Sciences Folks Foundation Medal. This conference's manifesto includes being outward-facing in seeking to build bridges with academia and other institutions. Muz's career is an excellent example of bridge building, and her own lab now increasingly has RSEs at the heart of it. Now that Muz has the unique position of leadership at Wellcome Sanger Institute, she can bring her perspective of how RSEs might become central not only to lab research visions, but the research visions of institutes and universities. Please join me in welcoming Muz to the stage.
Thank you very much, Simone, for the introduction, and Dave, what a fantastic conference that you've organized. I know you've done it for many months now, and so I've learned how to organize a conference from Dave, and I also learned how to give a great welcome, conference start speech from Dave, and I'm constantly learning from Dave. So this is a great pleasure to be here. I've never ever spoken in front of research software engineers, and after hearing Anne-Marie, I'm kind of like now also wanting to be a research software engineer. She talked about taking apart VHS. I'm going to focus a bit more on taking apart the human body. So there's going to be a bit of a gear shift, but there are lots of lessons that I've learned. And I was so struck by the kind of like save your work, because this talk, I lost it this morning. So, you still iterate and you're still learning at whatever age you may be. So I'm going to talk about, kind of my experience of how I've interfaced with research software engineers and research software engineering, and how I really think it's really important in terms of accelerating the translation of biomedical research. Technology is important. We heard about why technology is important, but technology is important in medicine, and some of the transformative technologies, for example, DNA sequencing, led to our understanding of the human genome. So this was the Human Genome Project, which essentially gave us the book of life, the complete DNA sequence of human. And what you then understand is that the variations in the human genome can actually predispose you to disease, etc. But we have this kind of like amazing observation in terms of all of us. We, in every single cell of our body, we have one genome. But the human body is extremely complex, and it's made up of up to 37 trillion cells. And how does this one genome encode for all of the cells in the human body? And the way that happens is that the different bits of the genome is active in individual cells. So this then results in gene expression being different in the individual cells, and gives rise to the identity of each of those cells. So your eye cells have a different gene expression to the hair cells and the liver cells, and so on and so forth. But it was only a few years ago that this kind of like resolution revolution happened in terms of being able to measure this at single cell resolution. So you could now look at gene expression in individual cells to really be able to kind of like work out what are the different cell types. And in addition to looking at gene expression, you now have like layers of information. You can measure proteins, you can measure some aspects of the DNA, and you can also, measure kind of like receptors that are on cells. But this requires you to kind of like take apart the body or the tissue, so you have to kind of like imagine kind of getting out the cells which are normally in a tissue context. And then there was another revolution in terms of being able to interrogate this in situ in the tissue. So you no longer have to split apart the cells and measure them. You actually know where you are. And that's really important because cells don't exist in isolation, but they exist within tissues. And there are many ways of doing this, and you can measure that information. And you can start to see how this numbers of different types of measurements come together, and we have to now put that information together in terms of integrating the different data modalities. So you have lots of data, different types of data, integrating all of that modality to kind of like reconstruct what a human body is made up of. And these are some of the cell atlasing technologies, and part of the technologies that drive this global initiative called the Human Cell Atlas, which is still ongoing, which is now going to define the book of cells of the human body. And unlike the Human Genome Project, representation and inclusion and diversity was kind of like built in from the very onset. In terms of Human Genome Project, is made up of a very small number of individuals who contributed their sample and the genome. But here what we really want is that this is going to be a globally representative atlas of the human body, and having researchers around the world, and also representation from cells, from individuals from around the world. And the idea is that if you have this reference, cells of the human body, you can use it as a guidebook and a blueprint to then ask the question, what happens during disease? If we now know what are the cells in the body, can we engineer and make those cells, not just individual cells, but also start to build tissues, and then that's called an organoid. But really, this is kind of like a whole lot of data, which gives you a phenomenal amount of resource. But it's very important to be able to kind of extract knowledge and meaning. And I'll give you some of the examples to show what can be extracted. So for example, the data from the gene expression profile of all the cells, you can interrogate that to look at which cells are expressing viral entry receptors. These are the surface molecules that a virus needs to attach itself in order to make its way into the cell and essentially cause an infection. And that data we used to find out where the receptors were that were enabling SARS-CoV-2, or what caused COVID-19. And that told us that it was actually not just the nasal cavity or the oral cavity, but the conjunctiva and cornea, and the importance of kind of like protecting the eyes. And this was really helpful information in terms of supporting why we should be wearing masks and limiting transmission. Another example is where a lot of the work that we've done looking at how the immune system develops, in utero, and these are the cells that are then possibly malfunctioning in the case of inborn errors of immunity. So you can now look at specific genes and where they may be expressed, and I've shown one very specific example. Each dot is an individual cell. The bit that's kind of like green is where that gene is expressed, and here I've shown GATA2, and this is exactly the type of data that you can extract from that information, which will then tell you what the patient may have. And in this context, it expressed in cells that make platelets, or cells that help you blood clot. And in these patients, they have a problem with clotting. You can also use these data sets to look at where drugs may be interacting, which cell types, and therefore what could be effective treatments for specific diseases, and also what are the side effects. And again, use this data to actually obtain information in terms of how we can make cells from scratch. Here you can make T cells, which are lymphocytes that are important in killing cancers and protecting you against infection, but also start to make tissues. Here is a skin organoid work from our own lab. And what you see on the right-hand side in terms of the streakiness is hair. So these are hair follicles growing in skin organoids. So you can just imagine the kind of like rich amount of information that you can get from these types of data sets. But there is a problem in terms of often what is shared are raw data. And that's the kind of like visible data that is, available for people. And what you then need to have is computing infrastructure and the expertise in terms of data analysis and coding skills. So really very few people benefit from this data, and not many people can extract the information, that rich information that's there. And those are the hidden insights, which are currently locked. And imagine the future of where all of this data can be valuable and available for people globally if you just need an internet, a computer, or a smartphone. So moving away from really needing complex coding skills to literally being able to use an app on your smartphone. And that's what I felt as a clinician when I kept pestering my team members to actually say, can you look this up, can you look that up? And really the story began with one of the team members sort of creating me a user interface and saying, go and do it yourself and leave me well alone, I've got better things to do. So I thought this was really revolutionary. It really unlocked, things for me. And I now feel that this is the way we're going to be doing it moving forwards. This is how society is going to benefit. And this is how there's going to be a global impact in terms of how that data can make a difference. So let's democratize data to accelerate clinical translation. We do very well data generation. We do very well bioinformatics computation. And I think this is where my personal view for the type of work that I do, how research software engineering can really make a massive difference. Data access, ensuring that it's fair and equitably accessed. And this is really important. But another thing that I kind of want to bring here, and we'll focus on in terms of some of the examples with regards to data formats and primarily focusing on point-and-click interfaces, is the kind of like ecosystem of how people work. In the past, researchers have mainly focused on either data generation and worked separately for bioinformatics and computation. And then you have like bioinformatics support unit or core facilities, and it's never quite integrated. You kind of like go with your data and then say, analyze my data. And to some extent, this can also be the case for research software engineering. And I see an ecosystem that's much more fluid, much more diverse, where all of these individuals with different expertise coming together in one kind of like a group or team, or maybe several teams. And this is really how we can kind of like drive things forward, because you're not just going there with an, saying, can you make this into an end product or just put it up as a user interface or whatever. And so my aim was to kind of like, develop some of this. It was really hard to get funding. I also didn't know where to go, but I was really lucky. I met somebody who knew Mark Turner at a dinner, and the next thing I knew, I had a chat with Mark and kind of like said, this is what I'm very interested in doing, and how do we make this happen? And he's been absolutely fantastic in making it happen. And currently in our team, we have 3 team members. Dave, whom you've known, and Daniela Basuto-Lozada, who's also here, and Luis Grimble, who will probably be online. And you can see the kind of like background with very different expertise and the kind of work that is ongoing in terms of the research. So this is really quite, speaking different languages. But the value of all of us working together is absolutely fantastic. And the interface with the bioinformatics and IT is crucial. So there are several things that research software engineering champion within our broader team, including training pipelines, and one that I would like to focus on is web portals. So this is how we really provide the data for the broader public, well beyond what is requested by journals in terms of just putting raw data. So every study that has come, and I have to acknowledge that a lot of these studies have been funded by Wellcome. And here you can see where we've got these, the kind of like summary of the study, what type of data is available, and the code that has been used to analyze the data and the findings. And I think this is really one way of making that data accessible. And also you've got these user interfaces, and I'm just going to show you, I showed this screen before, but the backstory for why I'm showing this is we provided that data, and one day I got an email from a researcher at the NIH saying, would you mind, can I use a screenshot from your web portal, please? Because it shows you the expression of the genes, and I now understand why my patients have clotting problems. And I'd like to include that in a textbook. And this is the kind of empowerment, and this is the kind of how we actually make a difference in the clinic and for the wider population that I think research software engineering can really play a role. And I mentioned about the data now being far more complicated in terms of the different modalities. And what we've been able to do is to now integrate the different types of data. So you don't just see the data as individual cells in a kind of like a two-dimensional rendering, but you can actually see where the cells are in situ. So this is a limb, and you can see the different cells and the genes that they express. And this is Web Atlas, which allows you to kind of like view in an integrated manner, which was developed together with the Omar Bayraktar's team and Tong Li here. So this is just kind of like a quick preview of what Web Atlas. So these are the three modalities. So each time you look at data in one modality, you can actually kind of like visualize it across all of the three. And you can zoom in and zoom out. And I don't really want to focus that much, but to say with Web Atlas, we can now integrate it with a clinical database called Decipher in the case of developmental disorders. So this is a very specific example. We've got human developmental data sets. We now pair up with an existing database that's hosted in EBI, the Decipher database, where patients actually submit their their criteria of the different types of diseases that cause developmental disorders, and we can now use that data to be able to say how that impacts the clinical manifestation of these disorders, so that clinicians from around the world can actually use this database. So here's the expression of genes that are involved in upper limb developmental defects, and it localizes to where the upper limb should be. And then you have all of the information about the disease, and you can also look at other genes, for example, that would be involved in kind of lower limb, and it would highlight where the expression of those genes would be. So these are the kind of things that really, will take the important insights from the large data generation efforts that are ongoing around the world to the clinic. So this is now the gene that's involved in lower limb. So what I guess I want to finish with is sort of talk about how the future aim with research software engineering with regards to the type of research that we're doing in developing this kind of like cell navigator where we bring together all of these different modalities and allow people to kind of like do like a Google Earth search, zooming in and zooming out through the human body, and actually also be able to kind of like query this database and using ML AI methods to get much more out of it. But it's really important to then have the expertise from this community to contribute to such an effort. And it doesn't have to be something that we start from scratch. There are a lot of existing infrastructure that's already there that can be leveraged to make this happen. And just to show the kind of like, archive data that is in EBI, the atlasing data and the software. And really to now be able to go from data, not just as a resource, but taking it for knowledge and meaning and making that impact to the clinic and beyond. And I'll stop there and just thank my team members and to acknowledge our funders and all of our collaborators around the world. And thank you very much for listening.
Thank you so much for a great talk, Muz. I think some questions will come through from the audience, so please send them in. But I can start off. I really liked what you said around the extraction of knowledge and meaning from data, and I'm sure a lot of people in this room focus on that problem quite a bit in their day-to-day. Specifically for you, it was around looking at inborn errors of immunity and things like this, but you really spoke about how computational people and RSEs unlocked this work beyond what journals were requiring. How do you see that that niche currently occupied in training pipelines and web portals extending out and having wider scope or larger scale when you look at institutes and universities, and what role RSEs can play, not just within sort of smaller teams, but across the wider ecosystem?
What I've given is kind of one example. There's a huge amount of how the narrative, we're coming back to telling narratives, of research can be. And one of the things that we've talked about is kind of having a platform that actually allows people to interact much, much more with the data in terms of not just static paper publication or print version publication, but going in and actually exploring data for themselves. And a lot of it is actually kind of good examples, test cases, and showing people how much this is actually going to kind of like benefit. And you can use this in education. Do you know what I mean? It's kind of like how much you can actually take it. And in terms of how we do this, I mean, personally, and also how I see it within Sanger, is to sort of build a research software engineering team within the program, and kind of like allowing this to interface, with EBI and outside. That's kind of a very specific example. But I think there's another component of not just having test cases and examples of how this is done, but also investment in terms of training. What could the training route look like for people who want to do research software engineering? The kind of heterogeneity in terms of what people do within what's called research software engineering, and having dedicated fellowships. And maybe this is where funders can come in and actually help that. And that's all.
Quite a lot of people are interested in the funding idea, actually. So the next question would be, how do we ensure funders are supportive of such infrastructures on a global scale?
We have funders here, so I think it'll be very interesting to hear their thoughts as well. It is a changing landscape. So when I first wanted funding for this, I didn't quite qualify to get research software engineering funding. That was one, and actually it was a philanthropic donor that enabled me to do this. I was really lucky. I just sat one dinner, person turned around and said, "What would you like?" And I said, money for research software engineers. That would be brilliant, because I was turned down saying that you can't ask for that money earlier that day. So serendipity, but in terms of kind of being nimble, just because it doesn't fit a particular category, and then being able to kind of like show what could be available. I mean, I think that's one way of kind of engaging funders. Maybe something that's any comments from funders could be helpful here as well. Putting you on the spot.
You're on the spot. Sure. We can move on to the next question. Is there a conflict between commercial funding, safeguarding patient privacy, and open data?
This is really interesting. A lot of this data is kind of anonymized data, so it's not something that can be attributed to the patient. And actually, one of the things we also undertook as part of the Human Cell Atlas was a big public engagement activity in terms of trying to ask people how they saw, the kind of like an entity that would have data that's kind of like available for people to kind of like browse and find out things from. And most people were actually really interested in kind of making their data available for greater benefit. So that was really interesting to establish and find out. But what I do feel we ought to be wary about is who then commercializes from the data and what you get from it. And that's something to think about. And there are many licenses for software. And I think I think this is something that I'm beginning to sort of carefully consider in terms of who benefits from this data, because research is changing, and there's a lot of biotech that's coming in using this data and then developing models, and models that may not necessarily be for the wider benefit. So there needs to be some consideration of how we engage and how we ensure that everybody benefits. And one way of doing that is to make sure that we don't just give the data, but we also work on the data to build these models.
Perhaps we'll do one final question so we don't overrun too much. But everyone, well, quite a few people were interested in the answer to this one. So many academics would love to be able to share their data in this way and perhaps have the expertise to develop these tools. The stumbling block in my experience is where should they be hosted and who should pay over the longer term. I've had a 10-year battle with my institution to find a solution to this problem with minimal success. 7 likes.
There are a lot of funder questions that are coming my way, and I'm kind of like almost putting it out there for the funders. And I guess a lot of this is depending on what type of data, who's going to benefit the data. There's a whole lot of discussion about who pays for data and then who maintains it. And actually, for all of these things, is there an exit strategy in terms of when do you actually stop making this available in the way that it is, because there isn't enough user usage or traffic. So I can't kind of comment in terms, I mean, I'm being more general rather than being very specific. But it's super important that these types of data is available for people, for everyone to benefit, and it should be kind of hosted in a way by organizations that would ensure that it is openly available and not just suddenly overnight that it becomes something that you have to pay for. That's something to bear in mind.
Thank you, Muz, and thank you for your really good talk.